Photosynthesis is a necessary process where light energy is converted into glucose, a sugar that powers cells. This process occurs in plants but also occurs in a couple species of animals. Photosynthesis consists of two cycles, the light cycle and the dark cycle, which is also called the Calvin cycle. Photosynthesis takes place inside chloroplasts, which are membrane-bound organelles found inside cells. In these two images, it shows examples of chloroplasts within plant cells. There can be anywhere from 20 to 100 chloroplasts in each cell. Chloroplasts were once an independent bacteria, but through evolution and endosymbiosis, the chloroplast has become a vital organelle inside the cell. That is why each chloroplast contains a double membrane. Each chloroplast contains thylakoids, stroma, and an inner and outer membrane. A stack of thylakoids is called a granum, and the inner space of the thylakoid is called the lumen. The light cycle occurs inside the thylakoid, also known as lumen, and also in the membrane or outer layer of the thylakoid. The dark cycle takes place in the stroma, the space in between the thylakoids, inside the chloroplast. The light cycle is a very important part of photosynthesis as it creates ATP and NADPH. These products are used to carry out the dark cycle. There are four important stages or parts of the light cycle, which includes photosystem 2, the electron transport chain, photosystem 1, and ATP synthase. This system is embedded in the membrane of the thylakoid. The light cycle begins in photosystem 2, when light from the sun or UV excites the electrons that are located in the photosystem. Photosystem 2 takes the light energy that was absorbed by chlorophyll. Chlorophyll are pigment molecules that are located in photosystems 1 and 2. Fun fact, chlorophyll absorbs most light except for green light, which is reflected, and why our eyes perceive most leaves as green. Photosynthesis can also only occur during the day as light energy drives the cycle. Continuing with photosystem 2, the energy from light photons excites electrons inside the photosystem protein. This excited electron splits water molecules into oxygen and hydrogen by borrowing its electron. This is called a redox reaction, as an electron is transferred from one molecule to another. The split oxygen molecules diffuse out of the cell, thus showing that oxygen is a product of photosynthesis, which is seen in the equation. The now excited electron leaves photosystem 2 and travels through the electron transport chain. This activates a hydrogen pump which moves hydrogen ions from outside of the thylakoid to the inside, increasing the concentration gradient by packing hydrogen ions inside the thylakoid. This electron is re-excited in photosystem 1 as light photons are received by the chlorophyll and use the light energy to re-excite the electron. Now, the electron converts NADP to NADPH through another redox reaction. The NADP molecule floats around the stroma and waits for the electron. NADPH is a crucial element to the function of the dark cycle. The final part of the light cycle is ATP synthase. The hydrogen ion gradient has increased inside the thylakoid through the splitting of water molecules in photosystem 2 and the electron transport chain pump. Next, the hydrogen molecules rush back out of the cell into the stroma through a protein called ATP synthase. The energy created from the rushing of hydrogen ions creates energy to convert ADP into ATP. ATP is the energy molecule of living organisms and is used in the dark cycle.
The dark cycle, otherwise known as the Calvin cycle or light-independent reaction, occurs in the stroma of chloroplasts, and the steps consist of carbon fixation, reduction, and regeneration. The Calvin cycle functions by using the NADPH and ATP produced in the light reaction in order to remove the carbon ion from the carbon dioxide molecule and convert it into a glucose molecule that can later be used in cellular respiration. In each cycle of the Calvin cycle, 3 carbon dioxide molecules are introduced in order to give the Calvin cycle 3 carbon atoms to work with. In the first step of the Calvin cycle, carbon fixation, 3 carbon dioxide molecules, CO2, are combined with 3 5-carbon ribulose biophosphate molecules, RUBP, in a reaction that is catalyzed by the enzyme Rubisco, which breaks off the oxygen from CO2 and binds the carbon in it to the RUBP in order to form 3 6-carbon molecules, which are then immediately split into 6 3-carbon phosphoglycerate molecules, PGA, as the 6-carbon molecule is unstable and PGA is stable. Those 6 3-carbon PGA then react with 6 ATP and 6 NADPH during the reduction reaction in order to turn into 6 3-carbon phosphoglyceraldehyde molecules known as pGal or G3P. The way that this reaction works is that the energy is taken from the 6 ATP in order to turn 6 PGA into bisphosphoglycerate and turn the 6 ATP back into 6 ATP and 6 inorganic phosphate. Then, the hydrogen is removed from the 6 NADPH in order to turn the bisphosphoglycerate into phosphoglyceraldehyde and turn the 6 NADPH into 6 NADP+ and 6 H2O. It is while it is in this state that one 3-carbon pGal molecule leaves the Calvin cycle in order to form glucose or other biomolecules. However, it is important to note that since each pGal molecule only has 3 carbon atoms in it, and 6 carbon atoms are required to form one glucose molecule, the Calvin cycle needs to occur 2 times in order to produce 1 carbon atom. The remaining 5 pGal molecules then react with 3 ATP in order to form 3 RUBP, which can then be used to repeat the Calvin cycle by binding to another 3 CO2 and going through the entire process again. This step of turning the 5 3-carbon pGal molecules into 3 5-carbon RUBP molecules is called regeneration and is carried out in order to allow the Calvin cycle to repeat itself countless times, and can be carried out since the 15 total carbon atoms from the pGal molecules are bound together until chains of 5 carbon atoms can be cut off from the chain and be formed into RUBP. This process also causes the 3 ATP to be converted into 3 ADP and 3 inorganic phosphates. Throughout the entire process of the Calvin cycle, 9 ATP and 6 NADPH are used and converted back into 9 ADP, 9 inorganic phosphate, 6 NADP+, and 6 H2O, all of which can then be used in the light reaction in order to repeat the initial steps of photosynthesis. There is also one 3-carbon pGal molecule that is released from this reaction every rotation due to the 3 CO2 molecules that are introduced to the cycle every time. Also, not every carbon that is put into the Calvin cycle is immediately spit back out in the form of pGal. Instead, it is more likely that 0 or 1 of the new carbons are spit back out in the form of a pGal. However, every carbon that is put into the Calvin cycle is eventually spit back out in the form of pGal.
For our experiment, we will be testing the presence of photosynthesis in spinach leaves. The materials you will need are sodium bicarbonate, or baking soda, liquid soap, a plastic syringe, leaf material, a hole punch, plastic cups, a timer, and light source.
The first step is to mix an 1/8 teaspoon of baking soda into 300 milliliters of water. This creates a 0.2% bicarbonate solution. Then mix a drop of soap into the solution and be careful not to create bubbles.
Then cut 10 discs from a spinach leaf. Put these leaves inside a syringe and pull a small amount of water into the syringe also. Using your finger, plug the hole at the top of the syringe and create a vacuum by pulling back on the plunger. Swirl the solution and the leaf discs around together. You will know that your leaves are ready to be put under light when you hold the syringe upright and the discs sink to the bottom.
Finally, put 3 centimeters of the bicarbonate and soap solution into a small plastic cup, then empty the leaf discs from the syringe and make sure that they all sink to the bottom.
The cup under a light. For our experiment, we used a grow light, but sunlight works too. Set a timer and watch how many leaf discs rise to the surface. Every minute, record how many leaf discs rise and stop the experiment after all 10 discs are floating.
The reason that leaves float most of the time is due to the fact that there are small spaces of air that allow the leaf's buoyancy to remain positive and allow it to float on water. Through the course of this experiment, we infiltrated those air bubbles within the leaf with a solution that had sodium bicarbonate in it because the solution both provides the carbon source for photosynthesis through the bicarbonate ion and allows the leaf to sink due to the air pockets being filled with liquid. Therefore, we can observe photosynthesis take place since when photosynthesis occurs, oxygen is produced and the pockets get filled up with that oxygen, increasing the buoyancy and allowing the leaf to float.